about my research, which I will today discuss with you on biobanking from an environment which is harshly or tremendously changing by climate change, so permafrost and glaciers. And I'm happy to discuss a little bit our research topic, and in particular our biobanking of microorganisms from these environments. These are a kind of research line. We are working with diversity, community profiling, and functional genetic potential in these fragile ecosystems. We are very much interested also in cold and stress adaptation, and in particular about microorganisms and their very particular genomic features in biotechnological application. We have a change in habitat and also a loss of habitat, for instance, vanishing glaciers or permafrost, which is thawing. And therefore, we also have a change in biodiversity or even biodiversity loss. So this is our motivation, that we study and preserve this unexplored microbial diversity in this cryosphere. So in Switzerland, we have two interesting monitoring systems. So this is the GLAMOS, which is just the monitoring the reduction or the decline of glaciers. And so we have 1,800 glaciers, and we know that they are tremendously disappearing or reducing. And in particular, in the last 10, 15 years, there was an accelerated decline. Also, we have the PERMOS, which is monitoring the ground temperature in high alpine locations in the Alps, so where we can see how fast the permafrost is thawing or how fast the active layer is increasing. So the permafrost is the continuously frozen soils. So from there we know that the permafrost is accelerated by thawing, and we know more or less what are the permafrost table in these high alpine sites. So we started to do a research on the permafrost and certain permafrost sites in the high alpine in the Swiss Alps, and there was a very surprising result, was that we had a huge diversity, microbial diversity in permafrost compared to active layer. We were thinking it's the opposite, that we have more diversity in the active layer than the permafrost. So the permafrost soils is roughly in the Switzerland, it's more or less in the Swiss Alps, roughly the carbon is 30,000 years old, so it's quite old soil. So we found also many unknown taxa in our metabarcoding study, and therefore we thought we should look more into permafrost microbiome and looking into this unknown taxa and their unknown metabolic potential. So we started to collect complex samples from these environments, either permafrost soils by digging into 1 to 2 meters of soils, or we are collecting subglacial ice or ice cores, or even we filtered hundreds of melted ice directly on the surface of the glaciers. So all these samples are stored in -20 in our freezers. And then we started to either two directions. Either we made some DNA-based studies, some metagenomics, or we started to isolate microorganisms from these environments on very oligotrophic medium. This means almost no carbon sources, no sugars, and at low temperature. So at 0 degree, we started to make isolations. So far, we have more than 1,500 bacteria and fungal isolates from permafrost and glacial ice. So we have we have a sample list of our microorganisms where we also have deposited some metadata, so from where it was isolated or collected and which medium it was used for isolation, what temperature GPS data where all these locations where we have isolated the microorganism. So many of them are unknown. We are still working on with taxonomists in collaboration to identify some of the unknown taxa. Here, two examples. From the Dammer Glacier in Switzerland, we found a new Glaciomonas, a new Chinos, and then also from Schafberg, so from a permafrost site, from ancient permafrost, we found a new yeast, cryophyllid yeast, Cryolionia schafbergensis. So these are just a few examples that we identify, but this is not our job. It's most in collaboration with taxonomists to identify new taxa. We also work together with artist, which was very fascinated about this old ancient permafrost and our isolates, and she was doing a lot of incubation experiment with different media, with different isolates, and then she photographed them over time to look at the grow forms and the grow style, and then this ended in a book called Unearthed. So now we have this 1,500 microorganisms in a biobank. So they're stored for long term at -80. And now we have two research directions where we're interested to get more information about the bioactive microbial gene products. So the bioactive potential of this microorganism, we are very much interested. And on one hand, it's more the kind of secondary metabolites, in particular antibiotic compounds. And the second research line is more on biodegradation of polymers. I quickly show a few examples from these two research lines. So now we have this 1,500 isolates. It's very obvious that we cannot test all of them in a screening system. So therefore we make first a pre-screening. And there we have wonderful genetic tools, genomic tools at hand, where we can make this pre-screening in this here for biosynthetic gene clusters. So gene clusters which encode for secondary metabolites like antibiotics, antifungal compounds. And from there, we after this pre-screening, we selected several hundred of our strains, and then we tested them in laboratory experiment as a kind of proof of concept. We tried to make confrontation tests. We looked into what they exude in a medium and characterized the medium. So some of them, they were very good or candidate for antibiotic producers. Here, for instance, in a confrontation test with a fungi, in the center, you see the fungi growing in one direction and then stopped, inhibited the growth from the bacteria on the border, on the left side, or on the top of the plate. So there we have interesting interesting degraders or producers of antibiotics. So the second research line we are following is so with our different expeditions we had in the Swiss Alps, but also in Arctic region like east of Greenland, Svalbard, or Arctic Russia. We collected a lot of plastic material in that area and the shores of these islands. And then we try to isolate them at a low temperature, and then we biobank them also. And now we have them in the biobank. We reactivate it, and we would like to see if they really are plastic degraders or they're just somehow surviving on plastic surfaces. And for this, again, we made a pre-screening for potential plastic degraders. We selected them by a plastic database, and then we made the proof of concept in lab experiment. We showed that some of the strains were able to degrade different polymers like nylon or polyethylene. And then we also sequenced all this genome from the potential plastic degraders, and there we find a kind of interesting taxa like Streptomycetes or the coccus, which seems to be quite strong in plastic degradation. So this is my conclusion. We have more than 1,500 strains biobanked from cold environments, and also complex and vital samples and DNA stored. So now they are ready to be further analyzed by their bioactive potential. And then we did some proof of concept where we looked how they produce antibiotics or can degrade plastics. Thank you very much for your attention.